So the first lab that we're going to be doing is the dialysis lab. The video for this lab will consist of two individual parts: the preparation for the lab and the analysis of the results. So our objective here is to distinguish between three types of the solutions: the true solution, the colloid solution, and the suspension. So true solution contains solute particles that are so small that they can pass through a membrane and a filter paper. For instance, solution of salt or glucose in water, or solution of ionic compounds in water. The colloid contains solute particles that are too large to pass through the membrane, but small enough to pass through the filter paper. Usually colloid solutions appear cloudy, but often you cannot discern individual particles. Examples of colloid solutions would be some solutions of protein in water, not all of them, or solution of a boiled starch in water. The suspension contains particles of solute that are so large that they cannot pass even through the filter paper, notwithstanding the membrane. If you will prepare the suspension eventually, the particles, the solute in the suspension will settle out of the solution, will precipitate on the bottom of the vial. Example would be sand and water, for instance, and as an example of a suspension. Now, membranes that we were talking about can be of one of three types. They can be permeable, any substance can pass through; impermeable, nothing passes through; or semi-permeable. They allow some substances to pass through, but not others. So cell membranes belong to the semi-permeable type of the membrane. They allow the diffusion of some parts, but they do not allow the diffusion of other particles. So we're going to be looking at the process of dialysis today. So we will look at the diffusion, in which very small molecules, well, in our case it will be ions, quite frankly sodium and chloride ions, and water, will pass through a membrane from the high to low concentration. So I often depict diffusion the following way. I think I did that before. It's a diffusion from high concentration to low concentration. It does not require energy, and this wedge-shaped structure represents the concentration gradient. So in the diagram here you can see the solution of sodium chloride is suspended in the dialysis tubing in the water. After a short period of time, the concentration of sodium chloride in the dialysis tubing becomes equal to the concentration of sodium chloride in the outside water in the beaker, because of the diffusion of sodium chloride from its higher to lower concentration. Over the materials first, we've got cellophane tubing, dialysis clips, three different solutions, the three beakers. Later we're going to do the test tubes, the Benedict reagent, all that kind of stuff. So the purpose here is to study the diffusion of particles through a non-living membrane to understand the diffusion in living cells. So we've got cellophane tubing. We moisten the tubing with the distilled water. So here's what we've got. So we've got three beakers with the distilled water. We've got cellophane tubing ready to go. And we've got graduated cylinders with the sodium chloride solution, boiled starch solution, and the dextrose solution. So now we're going to try and feel the cellophane tubing it seems like I have two pieces here. Okay. So this is how we folded and sealed the tubing. Look at this. I'm going to moisten the end of the tubing, and I'm going to rub the end between my fingers to open it up. So you see it's open now. And now I'm going to slowly pour the solution of So I've got my glucose in the tubing. Now I can fold another end, clip it as well, and put it in the beaker. So the entire solution is in the beaker. Then I'm going to do the same moisten it a little bit more. I'm going to do the same with the boiled starch solution. So you see I've got it in nice and well. And I'm going to clip it. And I'm making sure that this entire thing is in the beaker. And finally I do the same with sodium chloride. So I moisten the end the tube. I open it up. I pour my sodium chloride in there. And then I clip it. So it is now sealed, and I can place it there and wait for it to do the osmosis. So from now, that's going to be the number one part. In 30 minutes, we're going to do some analysis, whether or not the osmosis, the dialysis happened.